It's very nice to be here, and thank you for the invitation. And especially, it's very nice to meet great colleagues here. But what I want to speak about is an idea. It's an idea of an early warning system. So what I have for you here, it's a citizen science project, and these are getting very famous nowadays. But actually, you will see how efficient they can be. And this topic will be about ticks, of course. And what I want to speak first, why we speak about ticks. When we speak about ticks, it's not just about ticks. It's about all the nasty bugs which are coming to us, and they are bringing all the nasty diseases with themselves. So when we speak about ticks, we also speak about sandflies, midges, mosquitoes, whatever. And we speak about all these diseases which are increasingly coming in increasing numbers, increasing geographic regions, and all these things, and these are definitely trans-border problems. So this is not just a problem for Hungary, this is not just a problem for Serbia, so we have to get together sometimes and figure out some nice things, nice ways to tackle these diseases. What is waiting for us in the next, 5 years ago I was speaking about these slides okay, in 50 years, what will happen? And now I can tell you that in 10 years, what is coming. So if you just check the outside temperature, this is not a normal winter. If you remember this summer, we had a lot of very strange and sometimes dangerous weather patterns. And I have you this very nice publication which summarized all these effects of climate change. And the conclusion was that all these things which are coming, as we call climate change, these are facilitating mostly the vector-borne diseases and mostly the vectors of these diseases. So what is coming for us to get us in the next 10 years, or actually it's already here, are vector-borne diseases. And very tiny differences in average temperatures or whatever can be very impactful in different disease patterns. So I have this example of mosquito because I'm more like a mosquito guy than a tick guy. But what happened in West Nile virus situation? So just a tiny difference in the average temperature makes a lot of changes in the ecology of mosquitoes, birds, and finally it results in a more aggregated pattern of mosquito and bird, and finally in a very efficient extrinsic incubation situation for the mosquito. So finally we end up with more human cases. And this is again climate change. Tiny patterns in climate change can be very impactful in diseases. But let's get back to the original topic of tick-borne diseases and ticks. So Gabor Kemenesi, my dear colleague, figured out, some of you maybe already did this flagging method to collect ticks. So you are very familiar with this challenge. So this is extremely challenging. So Gabor Kemenesi, my dear colleague, figured out why to go to the field, why not to ask the general people to send us the bugs. Okay, let's do this. And we did this citizen science program, which I'm speaking now about, but we didn't want to stop here. So the idea was collecting all these samples, finding hotspots of important ticks. What is important tick for Hungary? Hyaloma, of course. We are waiting hyaloma to arrive, and you will see that it's actually here already. Finding hotspots of these ticks and get these ticks to the virus guy, which is me, and analyze these ticks, and let's see what we have here. So we can call this whole idea, this whole concept, as an early warning system. And as we see now, it's working. I won't speak about tick-borne diseases too much because you already heard these, but let's see this map of hyaloma. So Hungary was considered as the border region of hyaloma expansion. And of course, we have several publications from Hungary. Most probably those were dropped off from a bird or whatever, so they were found on cattle nearby the border. So we expected this is coming. And we established this nice citizen science program, and some of you are already involved in this cost action called Pragmatic. So the application for mobile phone is under development. So I expect this specific citizen science program to grow to a European-wide program. And this can be very, very useful. But now I present you the website. So what is needed for a good citizen science program? So how you move general citizens. You tell them, hey guys, you want to be a researcher? Okay, join, send your stuff. If you find this specific bug, just put it in an envelope and send it to us by post, or come into the research center and give it to us, and we will give you some nice answers. You need nice websites, of course, nice applications, mobile phone applications, things like this, and you need to tell general people what are you looking for. And as you can see, here is this nice example. You have to tell what is the hyaloma tick. For a Hungarian researcher, hyaloma tick is very strange. So we have dermacentors, which is like similar for the general eye, but hyaloma is so special, so we don't have anything similar in Hungary. And as you will see, we already have this. So when we started this program 3 years ago, and we received one hyaloma roofie pass in the first year. And as I told you, we don't just stop in receiving these specimens and putting the dot on the map. We start analyzing. So we put the question where we got this tick from. So we did this genetic analysis of the tick, and we saw that which cluster it's from. So most of you are very familiar with the transmission or the expansion of these ticks. So usually they are really traveling on birds. So bird migration routes can be a very important way of introduction of these ticks, but there are several other factors which can like put these ticks to another district or another country. So we were very interested in the origin of these specimens, and of course it was not just a roofie pass. It was we also received the hyaloma marginatum at the first year, and we did the same. So what I want to highlight here is these specimens can be very precious. So you don't have to spend your research money, go out flagging and trying to find these hotspots of ticks. You just need to ask general citizens, and they are very nice and send you these things. And what we found, and what I want to speak about, and what was shocking to us, we found multiple specimens after the year passed. So in the second year, we received more than 10 or nearly 15. We will see in the next slides how many, but we found also hotspots. So now we are contact with a cattle farmer, and whenever the cattle goes into the swampy region of the farm, they come out and they have a hyaloma marginatum. Okay, what the hell? So this is happening continuously. So we see now hotspots, and these hotspots cannot be found by a researcher just going randomly out to the field. So it's very useful to ask citizens. And this is more like my topic. This is virus topic. Of course, we analyze these specimens for viruses. What you do when you don't know what to find, of course, we screen them for Crimean-Congo hemorrhagic fever virus, because that's the first thing we do. They were negative so far, but we did also viral metagenomics, and we found very interesting things. And these are all the, like we can call the hidden virome of the ticks. So you can find very nice and very interesting viruses when you search these viruses. And we found this Wolsko tick virus, which is a Bunya virus. We don't know anything about this virus, but this was here, and this was introduced along with the ticks. So that's why a complex early warning system aided by citizen science is very important, because you can be one step forward to these things. So maybe this Wolsko virus will be important. We don't know, but we know now for sure that this is here in these ticks. We know the sequence. We know a lot of other details. So this is what we call outbreak prevention, or so we are one step ahead of these things. So what we have and what we received, you can see here that it's going very well. So the first year was 2021. We received 2 samples and then 9, and this year they are still in process. But what you can see, we have these hotspots in the middle of the country, and that is the hotspot of the cattle farm. And then we have another hotspot, if you are familiar with the Lake Balaton. This is more like to the northwest. Also, we have a hotspot there, again a cattle farm. So it seems that these cattle farms are very nice stepping stones to introduce this tick. And what we did, you already heard about the microbiome of ticks. Of course, when you ask citizens to send you hyaloma ticks, every tick will be hyaloma tick. So we receive a lot of Ixodes and other ticks, but these are also valuable specimens. So what we did, we analyzed these other tick species, and we wanted to try some new methods. We always looking in the future, and we always try new methods like faster, more deployable, more with less reaction time. So, and this nanopore sequencing is a very good way to get an idea about the microbiome of the tick. So you can do this kind of experiment in hours and you can get fast results. And this can be actually very important for clinicians. So if you have a patient and you don't want to do like, I don't know, 15 different PCR tests, of course the price is different, but the price is getting very nice, I can tell you. So in the last 5 years, it's getting lower and lower, but you can have a general idea what's in that tick. And if you see like a pathogen pattern or a candidate for a pathogen which can be linked to the symptoms, that can be very useful. And why I came here, that was two reasons. One reason was showing you this nice system of how you can use citizens, how you can get nice samples, and with a good laboratory backup, you can analyze these samples and get very nice scientific results. And still you are one step ahead of outbreaks. And the other reason was introducing ourselves. Yes. So we are very close to you, and some of you is already working with us. I just wanted to introduce our lab. We are in Pécs, which is very close to our Croatian colleagues and very close to Serbia. And all of you are very welcome anytime if you want to visit us and check our laboratory. If you are specifically interested in biosafety laboratories, we have a biosafety 4 laboratory with a training space. So if you are interested in or want to develop your skills in biosafety, you are more than welcome anytime. And of course, if you are interested in different collaborations with zoonotic pathogens, ticks, or not just ticks, you are also very welcome. So thank you very much.